Ancient viruses in our DNA, the key to gene regulation. Ancient viruses, the hidden architects of our DNA. A groundbreaking study reveals that ancient viruses, which have become part of our DNA, play a significant role in regulating the activation and deactivation of our genes. This fascinating discovery sheds light on how nearly half of the human genome is composed of segments known as transposable elements, TEs, often referred to as jumping genes, due to their ability to move around within the genome. These TEs are remnants of ancient viral infections that have been inherited through generations, dating back millions of years. For many years, scientists believed that these transposable elements were merely junk DNA, serving no real purpose in our genetic makeup. However, this new research challenges that notion, suggesting that these seemingly inactive segments of DNA are, in fact, vital for regulating gene expression, particularly during the early stages of development. The findings were published on July 18th in the journal Science Advances. Hiromi Nakao-Inoue, a research coordinator at Kyoto University's Institute for the Advanced Study of Human Biology and co-author of the study, stated, "Although our genome was sequenced long ago, many of its components remain a mystery. Transposable elements are believed to play crucial roles in the evolution of the genome, and their importance will likely become clearer as research progresses." Evaluating the role of TEs. The classification of TEs as junk stemmed from their perceived lack of relevance to protein synthesis, the essential molecules that construct and maintain cellular functions. While genes provide the instructions for protein creation, these repetitive transposable elements were long dismissed as non-functional. However, recent evidence suggests that these repetitive sequences are integral to gene regulation. For example, they often contribute to the production of non-coding RNA, a type of molecule that can influence other genes, aiding in cell differentiation and regulating embryonic growth. The advent of CRISPR technology has further enhanced our understanding of transposable elements. This revolutionary gene editing tool allows scientists to explore how TEs affect chromatin structure, the combination of DNA and proteins that form chromosomes, and can even activate gene expression in embryos shortly after fertilization. In this study, researchers concentrated on a specific group of TEs known as MER11, which are part of a larger class that entered primate genomes approximately 40 million years ago. They categorized the sequences within the MER11 family based on their evolutionary relationships, resulting in 4 subgroups ranging from MER11G1, the oldest, to MER11G4, the youngest. To investigate the impact of these TEs on cellular function, the team introduced nearly 7,000 sequences into laboratory cell cultures. These sequences, derived from humans and other primates, were inserted into stem cells and early neural cells, allowing the researchers to measure gene activity. The results indicated that the youngest members of the MER11 family, specifically MER11G4, exhibited a remarkable capacity to activate genes. They contained unique transcription factor binding sites, specific DNA motifs essential for development, that serve as docking points for proteins regulating gene expression. Diversity among species. Interestingly, variations in the MER11G4 sequences were observed among humans, chimpanzees, and macaques, with these differences altering the regulatory effects of the sequences across species. Cristina Tuffarelli, a geneticist at the University of Leicester's Cancer Research Center, who was not involved in the study, remarked, "This research underscores the vast amount we still have to learn from the genome, particularly regarding virus-like transposon repeats, whose diversity has often been overlooked." She emphasized that this work opens numerous pathways for future research, suggesting that similar approaches could be applied to other transposable elements, potentially revealing more about their regulatory functions. Tuffarelli also noted that future studies might involve using CRISPR to delete specific TE segments, helping to clarify their roles in gene regulation in both healthy and diseased states. Ben Turner, a staff writer at Live Science, covers a range of topics including physics, astronomy, technology, and climate change. He holds a degree in particle physics from University College London and has a passion for literature, music, and chess. Unlocking the secrets of viral DNA, how it regulates our genes. What if the so-called junk tucked deep within your DNA was actually crucial for what makes you, well, you? For years, fragments of ancient viruses embedded in our genetic code were written off as useless leftovers from our evolutionary past. But new research is now turning that assumption upside down. These viral sequences, comprising nearly 8% of our entire genome, might play a surprisingly active role in regulating how our genes function. And that's not just a minor tweak. It reshapes the way we understand human biology itself. But here's where it gets truly fascinating. An international team led by Guillaume Bourque of McGill University, together with colleagues at Kyoto University, recently published a groundbreaking study in Science Advances. Their research, titled A Phylogenetic Approach Uncovers Cryptic Endogenous Retrovirus Subfamilies in the Primate Lineage, introduces a fresh way to trace the evolutionary history of viral DNA scattered across our genome. Instead of relying on traditional annotations, which may miss subtle differences, they used a phylogenetic approach. By comparing hidden viral sequences across species, the team managed to expose a previously undetected subfamily dubbed MER11G4. What's so special about MER11G4? It seems to be especially active in human stem cells and those of our close cousins, chimpanzees, hinting it could be a master regulator in both species. Here's the part that challenges what most people take for granted. The current maps that scientists use to identify viral DNA in our genome shouldn't be considered complete or final. It's time to revisit and refine them, urges Professor Bourque. Their findings suggest some viral fragments might still be influencing which genes are switched on or off, even influencing our health, susceptibility to disease, or what fundamentally differentiates humans from other primates. This raises a host of exciting, perhaps even controversial questions. Could so-called junk DNA be a key driver in evolution or even personalized medicine? How might our understanding of inherited disease shift if these viral remnants are playing unexpected roles? The research, supported by a network of Canadian and Japanese scientific organizations, opens the door for future discoveries that could reshape fields from genetics to medicine. But it also leaves us with a challenge. Should we be rethinking how we look at the very building blocks of life? Or are we reading too much into the traces of ancient viruses in our genome? This debate is far from over. What do you think? Is it time for the scientific community to overturn old assumptions about junk DNA? Or does more evidence need to come to light? Share your perspective. Do these findings excite you, or do you remain skeptical? Drop your thoughts and join the conversation below. Scientists discover secret code in human DNA, unlocking the mysteries of our DNA, a surprising discovery. In a groundbreaking revelation, a diverse group of scientists has uncovered that segments of what was once dismissed as junk DNA in the human genome actually hold significant importance. This finding challenges the long-held belief that these sequences serve no purpose, suggesting instead that they play crucial roles in our genetic makeup. Published in the journal Science Advances. This study delves into transposable elements, TEs, a category of DNA sequences capable of relocating themselves within the genome through a biological copy and paste mechanism. Remarkably, these jumping genes constitute nearly half of human DNA, and their presence is even more pronounced in other organisms. The research team, hailing from Japan, China, Canada, and the United States, focused on a specific family of TEs known as MER11. They discovered that these sequences can significantly influence gene expression, acting as genetic switches without altering the actual DNA structure. Fumitaka Inoue, a co-author of the study from Kyoto University, emphasized the mystery surrounding our genome, stating, "Our genome was sequenced long ago, but the function of many of its parts remains unknown. This highlights the ongoing quest to understand the complexities of our genetic code. The MER11 sequences are classified as long-terminal repeat, LTR retrotransposons, which are believed to have originated from an endogenous retrovirus, ERV, that infected a common ancestor of primates millions of years ago. This virus managed to integrate its genetic material into the host's DNA, resulting in a legacy that persists today, with researchers estimating that at least 8% of the human genome is derived from these retroviruses. The presence of TEs, including MER11, creates a complex puzzle for scientists to decipher. The authors of the study argue that existing methods for classifying and annotating TEs are flawed, often leading to the misclassification of these sequences as mere genetic debris. This realization prompted them to develop a new classification system. They stated, "The proper classification and annotation of LTR instances is critical to understanding their evolution, co-option, and potential impact on the host." Using their innovative system, the researchers categorized MER11 sequences based on their evolutionary relationships and their preservation across primate genomes. They further divided MER11 into 4 distinct subfamilies, labeled MER11 G1 through G4 according to their age. This classification enabled the team to compare the MER11 subfamilies with epigenetic marks, chemical modifications that can influence how proteins function and consequently gene activity. Notably, these epigenetic marks can modify cellular behavior without changing the DNA itself, such as silencing a gene that should be active. Establishing a connection between the MER11 subfamilies and these markers is essential for understanding their role in gene expression. In their investigation, the team analyzed approximately 7,000 MER11 sequences from humans and primates, assessing their impact on gene activity. They found that the youngest subfamily, G4, exhibited a remarkable capacity to influence gene expression. This influence is attributed to unique DNA motifs within G4 that attract transcription factors, proteins that regulate gene activation and deactivation. Lead author Xun Chen from the Chinese Academy of Sciences noted, "Young MER11 G4 binds to a distinct set of transcription factors, indicating that this group gained different regulatory functions through sequence changes and contributes to speciation. The implications of these findings are profound." What was once considered junk DNA has evolved to play a vital role in gene regulation, hinting at a rich and largely unexplored evolutionary history. Inoue remarked, "Transposable elements are thought to play important roles in genome evolution, and their significance is expected to become clearer as research continues to advance." What do you think? Are we on the brink of a new understanding of our genetic heritage? Or is this just the tip of the iceberg? Share your thoughts in the comments below. For more intriguing insights into genetics, check out how Elon Musk's eugenics startup is examining DNA to assess potential intelligence in future generations.